Welcome friends to a new session on molecular biology. In this session, we will discuss the initiation, elongation and termination of transcription in eukaryotes. In prokaryotes and eukaryotes, the process of RNA synthesis is similar, but the process is considerably more complex in eukaryotes. In eukaryotes, RNA is synthesized in nucleus and must be transported to the cytoplasm for translation. In prokaryotes, mRNAs often contain the coding regions of two or more genes. Such mRNAs are said to be multigenic, that is, controlled by multiple genes. On the other hand, many of the eukaryotic transcripts that contain the coding region of a single gene are monogenic. In eukaryotes, the bulk of primary transcripts in a nucleus are called heterogeneous nuclear RNA, that is hnRNA, because of the considerable variation in the sizes of RNA molecules present. Majority of these hnRNAs are non-coding intron sequences which are excised from the primary transcripts and undergoes various processes before leaving the nucleus. The RNA transcripts are coated with RNA binding proteins during or immediately after their synthesis in eukaryotes. These proteins protect the gene transcripts from degradation by ribonucleases, that is, enzymes that degrade RNA molecules. The average half-life of E. coli is less than 5 minutes. The average half-life of a gene transcript in eukaryotes is about 5 hours. The RNA molecules that are translated on ribosomes are called messenger RNAs, that is mRNAs. During transcription, one strand of gene is used as a template to synthesize a complementary strand of RNA is called the gene transcript. In prokaryotes, the primary transcript is equivalent to the mRNA molecule. In eukaryotes, the primary transcripts must be processed by the excision of specific sequences and the modification of both termini before they can be translated. General features of RNA synthesis. RNA synthesis occurs by a mechanism that is very similar to that of DNA synthesis, except that 1. The precursors are ribonucleoside triphosphate rather than deoxyribonucleoside triphosphates. 2. Only one strand of DNA is used as a template for the synthesis of a complementary RNA chain in any given region. And 3. RNA chain can be initiated de novo without any requirement for a primer strand. The RNA molecule produced will be complementary and identical to DNA template strand, except that uridine residues replace thymidines to the DNA non-template strand. If the RNA molecule is an mRNA, it will specify amino acids in the protein gene product. This specification is accomplished by nucleotide triplets called codons. Each codon specifies a single amino acid in a polypeptide chain. Therefore, mRNA molecules are coding strands of RNA. They are also called sense strands of RNA because their nucleotide sequence makes sense in that they specify sequences of amino acids in the protein gene products. An RNA molecule that is complementary to mRNA is referred to as antisense RNA. The synthesis of RNA chains like DNA chains is in the 5 prime to 3 prime direction with the addition of ribonucleotides to the 3 prime OH group at the end of the chain. The reaction involves a nucleophilic attack by the 3 prime OH group on the nucleotidal phosphorus atom of the ribonucleoside triphosphate precursor with the elimination of pyrophosphate as in DNA synthesis. This reaction is catalyzed by the enzyme RNA polymerases. The overall reaction is as follows. Here N is the number of moles of ribonucleotides triphosphate RTP consumed. Ribonucleotides monophosphate RIMP incorporated into RNA and pyrophosphate produced. RNA polymerases initiate transcription at specific nucleotide sequences called promoters, which are different in prokaryotes and eukaryotes. RNA synthesis takes place within a locally unwound segment of DNA, sometimes called transcription bubble, which is produced by RNA polymerase. There are 4 types of RNA molecules. The 4 different types of RNA molecules play essential role in gene expression. We have already discussed messenger RNAs, the intermediaries that carry information from DNA to the ribosomes where proteins are synthesized. Transfer RNAs, that is tRNAs, are small molecules that function as adapters between the amino acids and the codons in mRNA during translation. Ribosomal RNAs, that is rRNAs, are structural components of the ribosomes, the intricate machines that translate nucleotide sequences of mRNAs into amino acid sequences of polypeptides. Small nuclear RNAs, snRNAs, are structural components of spliceosomes, the nuclear structures that excise introns from nuclear genes. Eukaryotic ribosomes are more complex than prokaryotic ribosomes. The 4 prokaryotic rRNAs designated as 5S, 5.8S, 18S and 24S rRNAs are approximately 120, 160, 1900 and 4700 nucleotides in length respectively. All 4 types of RNA, mRNA, tRNA, rRNA and snRNA are produced by transcription. Whereas a single RNA polymerase enzyme catalyzes all transcription in E. coli, eukaryotes ranging from the single celled yeast to humans contain 3 different RNA polymerases. All 3 eukaryotic RNA polymerases designated as RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. These enzymes are more complex with 10 or more subunits. All the eukaryotic RNA polymerases require the assistance of other proteins called transcription factors in order to initiate the synthesis of RNA chains. The RNA polymerase 1 is located in the nucleolus, a distinct region of the nucleus where rRNAs are synthesized and combined with the ribosomal proteins. It catalyzes the synthesis of all ribosomal RNAs except the small 5S rRNA. RNA polymerase 2 is located in the nucleus and it transcribes nuclear genes that encode proteins and perhaps other genes specifying hnRNAs. RNA polymerase 3 is also located in the nucleus, catalyzes the synthesis of the tRNA molecules, the 5S RNA molecules and small nuclear RNAs. The process of transcription in eukaryotes can be divided into 3 stages. 1. Initiation of RNA chains. 2. Elongation of the RNA chain and the addition of 5 prime methyl guanosine caps. And 3. termination of transcription by chain cleavage and the addition of 3' poly A tails. First, let us look into initiation of RNA chains. Unlike the initiation of transcription in prokaryotes, eukaryotic RNA polymerases cannot initiate transcription by themselves. All 3 eukaryotic RNA polymerases require the assistance of protein transcription factors to initiate the synthesis of an RNA chain. These transcription factors must bind to a promoter region in DNA and form an appropriate initiation complex before RNA polymerase will bind and initiate transcription. The common feature of this transcription, however, involves binding of a number of transcription factors to DNA sequences upstream of start point. Here, the structure of a promoter recognized by RNA polymerase II is shown. The Tata and CAAT boxes are located at about the same positions in the promoters of most nuclear genes encoding proteins. The number of transcription factors associated with the initiation process increases as the variety of genes transcribed by the polymerase increases. These transcription factors help in DNA binding of an RNA polymerase to constitute what is described as transcription. After the formation of this complex, initiation of transcription occurs. All non-transcription factors may not necessarily recognize DNA sequences, and a factor may instead recognize another factor or may even recognize RNA polymerase. The initiation of transcription involves the formation of a locally unwound segment of DNA, providing a DNA strand functions as a template for the synthesis of a complementary strand of RNA. The formation of the locally unwound segment of DNA required to initiate transcription involves the interaction of several transcription factors with specific cis acting sequences in the promoter region for the transcription unit. The promoters recognized by RNA polymerase II consist of short conserved elements or modules located upstream from the transcription start site is called Tata Box. The position is at +1. It has the consensus sequences TA, TA, AAA reading 5' to 3' on the template strand and is centered about at position -30. The Tata Box plays an important role in positioning the transcription start point. The second conserved element is the CAAT box, which occurs near -80 position and has the consensus sequence GG, CC, AA, T, CT. Two other conserved elements, the GC box consisting of GGG, CGG and octamer box consisting of A triple T, GCAT is often present in RNA polymerase II promoters. The CAAT, GC and octamer sequences influence the efficiency of a promoter in transcription initiation. The initiation of transcription by RNA polymerase II requires the assistance of several basal transcription factors. These transcription factors must interact with the promoters in the correct sequence to initiate transcription. The initiation of transcription in eukaryotes begins with the binding of TF2D to the Tata box. The key initial event in initiation is the recognition of the Tata box by the TBP Tata box binding protein a 30 kilodalton component of the 700 kilodalton TF2D complex. TBP is a saddle shaped protein consisting of two domains which are similar in structure. The Tata box of DNA double helix binds to the concave surface of TBP and induces large conformational changes in the bound DNA. The DNA is substantially unwound to widen its minor groove, enabling extensive contact of TBP. 4 phenylalanine residues are intercalated between base pairs of the Tata box. Next, TF2A joins to the complex, followed by TF2B. TF2F, an ATP dependent helicase, first associates with RNA polymerase II, and then TF2F and RNA polymerase II joins the transcription initiation complex. TF2F contains 2 subunits which has DNA unwinding activity. Thus, TF2F probably catalyzes the localized unwinding of the DNA double helix required to initiate transcription. TF2E join the other factors to form a complex called basal transcription apparatus. 2 other factors, TF2H and TF2J, join the complex after TF2E, but their locations in the complex are unknown. TF2H contain an enzymatic activity that phosphorylates RNA polymerase II. One hypothesis is that the phosphorylation of RNA polymerase II by TF2F releases the enzyme from the initiation complex at the promoter, so that it can move downstream and mediate RNA chain extension. Similar promoter complex or transcription factors are required to initiate transcription by RNA polymerase I and polymerase III. RNA polymerase I and polymerase III use some of the same transcription factors as polymerase II, and some factors are specific to each enzyme. The promoters of genes transcribed by polymerase I and polymerase III are quite different from the promoters utilized by RNA polymerase II. Elongation of the RNA chain and the addition of 5' methyl guanosine caps. Once eukaryotic RNA polymerases have been released from the initiation complex, they catalyze RNA chain elongation by the same mechanism as the RNA polymerase of prokaryotes. The most extensively modified transcription product is the product of RNA polymerase II, referred to as pre-mRNA. Most pre-mRNA molecules are spliced to remove the introns. Early in the elongation process, the 5' ends of eukaryotic pre-mRNAs are modified by the addition of 7-methyl guanosine, that is 7-mg caps. These 7-mg caps are added when the growing RNA chains are only about 30 nucleotides long. The 7-mg cap contains an unusual 5' triphosphate linkage and 2 or more methyl groups. First, a phosphate is released by hydrolysis. The diphosphate 5' end then attacks the alpha phosphorus atom of GTP to form a very unusual 5' triphosphate linkage. The distinct terminus is called a cap. The N7 nitrogen of the terminal guanine is then methylated by S-adenosyl methionine to form cap 0. The adjacent ribosomes methylated to form cap 1 or cap 2. The 7 mg caps are recognized by protein factors involved in the initiation of translation and contribute the stability of mRNAs by protecting their 5' ends from phosphatases and nucleases. Termination of transcription by chain cleavage and the addition of 3' poly A tails. As mentioned earlier, the 3' end of pre-mRNA transcript is also modified. The 3' ends of RNA transcripts synthesized by the enzyme RNA polymerase II is produced by endonucleolytic cleavage of the primary transcripts rather than by the transcription termination. The actual termination of transcription events often occur at multiple sites that are located to 1000 to 2000 nucleotides downstream from the site that will become the 3' end of the mature mRNA transcript. That means, transcription proceeds beyond the site that will become the 3' terminus and the distal segment is removed by endonucleolytic cleavage. The cleavage event that produces the 3' end of an mRNA transcript usually occurs at a site 11 to 30 nucleotides downstream from a conserved sequence consensus AAU AAA located near the end of the transcription unit. Cleavage does not occur if this or a segment of some 20 nucleotides on its 3' side is deleted. The presence of the internal consensus sequences AAU AAA in some mature mRNAs indicates that consensus sequence AAU AAA is only part of the cleavage signal. Its context is also important. After cleavage by the endonuclease, a poly A polymerase enzyme adds about 250 adenylate residues to the 3' ends of the mRNA transcript. The addition of poly A tails to eukaryotic mRNAs is called polyadenylation. ATP is the donor in this reaction. The formation of poly A tail on mRNA transcripts requires a specificity component that recognizes the AAU AAA consensus sequences in RNAs, binds to them and directs both the cleavage and the polyadenylation reactions. The specificity component, the endonuclease and the poly A polymerase enzyme are present in a multimeric protein complex that catalyzes both cleavage and polyadenylation in tightly coupled reactions. In contrast to RNA polymerase II, both RNA polymerase I and polymerase III respond to discrete termination signals. RNA polymerase enzyme 1 terminate transcription response to an 18 nucleotide long sequence that is recognized by an associated terminator protein. RNA polymerase III responds to a terminator in E. coli. The role of the poly A tail is still not firmly established. However, evidence that it enhances the stability and translation efficiency of mRNA is accumulating. It also plays an important role in their transport from the nucleus to the cytoplasm. Blocking the synthesis of poly A tail by exposure to 3' deoxyadenosine, that is codisepine, does not interfere with the synthesis of the primary transcript. mRNA devoid of a poly A tail can be transported out of the nucleus. The half-life of an mRNA molecule may also be determined in part by the rate of degradation of its poly A tail. An mRNA molecule devoid of a poly A tail is usually a much effective template for protein synthesis than is one with a poly A tail. Let us summarize the following discussion on initiation, elongation and termination of transcription in eukaryotes. Transcription involves the synthesis of an RNA transcript from complementary to one strand of DNA of a gene. The process of transcription in eukaryotes involves three stages: initiation of RNA chains, elongation of the RNA chain, and the addition of 5' methyl guanosine caps, and termination of transcription by chain cleavage and the addition of 3' poly A tails. Eukaryotic RNA polymerases require the assistance of protein transcription factors to initiate the synthesis of an RNA chain. Initiation involves the formation of a locally unwound segment of DNA, providing a DNA strand functions as a template for the synthesis of a complementary strand of RNA. In the elongation process, the 5' ends of eukaryotic pre-mRNAs are modified by the addition of 7-methyl guanosine, that is 7-mg caps. The addition of poly A tails to 3' ends of the eukaryotic mRNA transcript is called polyadenylation. I will give you certain assignments based on the discussions what we had. 1. Briefly explain the general features of RNA synthesis. 2. Write short notes on 4 types of RNA molecules which play essential role in gene expression. 3. Explain the process of transcription in eukaryotes. 4. Discuss the elongation of the RNA chain and the addition of 5' methyl guanosine caps. 5. Give a brief account on termination of transcription by chain cleavage and the addition of 3' poly A tails. I will also give you certain references for your further reading. 1. The book with the title Biochemistry 5th edition, authored by Berg, Timsko, Stryer in the year 2008 and published by Freeman and Company, New York, USA. A second book is with the title Cell Biology 7th edition, authored by Karp in 2014, published by John Wiley & Sons, Singapore. A third book is with the title Molecular Biology of the Cell 5th edition, authored by Alberts, Johnson, Lewis, Raff, Roberts and Walter in the year 2007 and published by Garland Science, New York and London. A fourth book is with the title Gene 9, authored by Levin in the year 2007 and published by Jones and Bartlett Publishers, Sudbury, Massachusetts. A fifth book is with the title Biochemistry, authored by Davidson and Sitman in the year 1997, published by Waverly, New Delhi. A sixth book is with the title Principles of Genetics, authored by Gardner and Nestad in the year 1984 and published by John Wiley, New York. A seventh book is with the title Principles of Genetics, authored by Sinnett, Dunn and Dobzhansky in the year 1958 and published by McGraw-Hill, New York. I will also give you certain websites for your further reference. The first one is www.bioencyclopedia.org. A second one is www.oxfordgenetics.com. We have come to the end of this session. We will meet in another session with a new topic. Until then, bye.